Hi kids, so now let us discuss the assays for your lactate dehydrogenase, including your acid phosphatase and your alkaline phosphatase. Now again, we haven't performed your LDH and your ACP, but then again, we will still be discussing their package inserts. And so I will be sending to you a PDF file on the package insert that I will be discussing here in our video lecture. So to begin with, we will start with your lactate dehydrogenase. So the EC number of your lactate dehydrogenase is 11127. And since we are talking about the first digit of your EC number, which is 1, so that means that it's under your classification of enzyme, which is oxidoreductase. And that classification can be found in your systematic name. So the systematic name of your lactate dehydrogenase is L-lactate NAD plus oxidoreductase. Now, the function of your LDH is to catalyze the interconversion of your lactic and pyruvic acid or your lactate and your pyruvate. And going back to how important this interconversion is, recall that whenever there is no oxygen or there is not enough oxygen in our body, this interconversion would take place. Yung glycolysis, yung citric acid cycle nyo, and even yung oxidative phosphorylation, all of those processes needs oxygen. Now, there may be cases that a certain individual would lack enough oxygen in order to pursue those processes. And when your glycolysis, your oxidative phosphorylation, and your citric acid cycle will not begin, then that would mean that there will also be a decline in your ATPs or your energy. So to continue providing energy to our body, what our body would do in cases if there is not enough oxygen would be this process, the interconversion of your lactic and pyruvic acid. So the role of your LDH here is to transfer hydrogen. That is why your coenzyme NAD is needed here, because that is where your oxidoreductase, your oxidation reduction will take place. So looking at your chemical reaction here on your left side, here's NAD. And then ang mangyayari po, it will become your reduced nicotinamide adenine dinucleotide. And another hydrogen ion will be removed. Saan po galing yung another hydrogen ion? So it comes from here. And yung isang hydrogen ion is here. So now let us go to the assays. Now, for your lactate dehydrogenase kids, again, your lactate dehydrogenase also has their own, or it has 5 different isoenzymes, and these are your LD1 until LD5, and that each of your isoenzymes have 2 subunits, and these are your, diba, your letter H and your letter M. So whenever we try to analyze isoenzymes, so your reference method will always be your electrophoresis. Now, lactate dehydrogenase also has presence of atypical bands, or what we call your abnormal isoenzymes, and that is your LDH being in complex with your immunoglobulin. Now, what specific type of immunoglobulin it would be? So that would be your IgA or your IgG. So upon checking your electrophoresis, this lactate dehydrogenase, which is in complex with your immunoglobulins, will be found to migrate in between your LD3 and your LD4. Now, the good thing about this atypical band, the good thing about this abnormal fraction is that it is not associated with any clinical abnormality. And then after we separated your different isoenzymes, this will be subjected to your fluorometry and your colorimetry, so that we'll be able to detect their migration and which portion are they in our columns. So other assays that we can do, especially in terms of isoenzyme separation, would be your immunoinhibition, your chemical inhibition, and in terms of the differences in substrate affinity. So in talking about differences in substrate affinity, we can make use of a different substrate here in our lactate dehydrogenase assay, and that would be your alpha hydroxybutyrate. Now, these substrate kids have increased affinity to your H subunit. So, going back to the different isoenzymes of your LDH, ito po yung sinasabi ko kanina, they are composed of two subunits, and that is your letter H and letter M. Now, since your LDH1 have the most number of H subunit, then therefore, in terms of this assay, it's more on trying to measure the activity of your LD1. And the enzyme that would represent your LD1 activity would be your alpha hydroxybutyrate dehydrogenase. Again, this is your enzyme, and this enzyme is considered to be a representative of your LD1 activity. So, kanina po, ito pong nasa taas, it's just your substrate. What we will measure is the activity of your enzyme, and that enzyme is your alpha hydroxybutyrate dehydrogenase. So, that enzyme is a representative of your LD1 activity, but just take note that this is not specific. Why hindi po siya specific? It's because looking at your other isoenzymes, they are not devoid of your H subunit with the exception of your LD5. Kasi diba si LD5 lang po yung may 4 na M subunit, but the rest, including your LD2, LD3, and LD4, they still have your H subunit, thus making this test not specific. So, lactate dehydrogenase assay also has the same reaction methods like your creatine kinase in terms of having or being able to measure. We could actually measure its forward and reverse reaction, but always, kids, your reverse reaction will always be faster and will always be having lesser interference. But the problem with the reverse reaction here of your lactate dehydrogenase is that it is more susceptible to substrate exhaustion and loss of linearity. But then again, upon looking at your package insert, especially your Agape na brand, they still make use of your reverse reaction. We just need to make sure that there is enough substrate so that there won't be any loss of linearity in our processing. So, the optimal pH for your forward reaction is 8.3 to 8.9, and the optimal pH for your reverse reaction would be 7.1 to 7.4. Now, the reverse and the forward reaction of your LD also have their specific names. For your forward, that is what we call your Walker method. For your reverse, that is what we call your Wroblewski-Ledoux. Although I'm not quite sure if I pronounced it right, then again, this is how you spell it's the name of the method. So again, for your forward reaction, that is your Walker method. For your reverse reaction, that is your Wroblewski-Ledoux. So going back to your chemical reaction, so again, this is representing your forward starting from your lactate. If it is the reverse, then we will begin with your pyruvate. So just correct the spelling of your pyruvate here. So how about the sources of error of your lactate dehydrogenase? Now again, lactate dehydrogenase have a high concentration of your red blood cell has a very high concentration of your lactate dehydrogenase, then therefore, bawal po ang you-hemolyze sa sample. RBC contains LD concentration around 100 to 150 times than found in your serum. Now, LD activity isn't stable in your serum regardless of temperature. So, meaning to say, kids, your sample should never be refrigerated. It should only be stored at room temperature and should be analyzed within 48 hours. Now, why hindi po siya pwede ilalagay dun sa refrigerator po natin? It's because of the presence of your LD5. Now, LD5 is your most labile isoenzyme. And when we say most labile, meaning to say it is easily destroyed, meaning to say it will easily lose its activity more quickly if it is placed at your ref temperature than at room temperature. So LD isoenzyme analysis should be stored at room temperature and analyzed within 24 hours of collection. So now we go to discussing your package insert. Now, looking at the principle of your lactate dehydrogenase, again, we are using your reverse reaction because we started with your pyruvate and looking at the product, which is your lactate. Now, the same as what we did all the time whenever we process your enzymes, we will always be given two reagent, and in preparing your reagent, that is a 1:5 dilution, placing 1 volume of your reagent 2 and 4 volumes of reagent 1. Now, for the reagent storage and stability, it is stable until the indicated expiry date as long as it is stored in your ref and protected from light. So for the interferences, kids, as you can see, wala pong nakasulat. There is nothing about hemoglobin because here, even as low as 1 mg per dl, bawal. There should really be no hemolysis. There should really be no presence of hemoglobin. It should really be devoid of any red blood cells. So for your test parameters, as written here, of course, this is a kinetic method. And what you will notice in terms of your slope, upon reading the absorbance per minute, it would be decreasing. The wavelength is 340 nm, and our temperature here should be 37 degrees Celsius. And then, number of readings is 3 times every minute. So, interval is 60 seconds. Your sample volume is 10 microliter, and your reagent volume is 1000 microliter. Now, if you wanted to convert your U per liter to your SI unit, which is your microcat per liter, then you need to multiply it by 0.017. So for the procedure, we've made mention about this a while ago, nakalagay po dun sa test parameter. So again, we need to make your working reagent first, and once we already mix thoroughly your working reagent, get 1000 microliter of it, place it in another test tube, and then add 10 microliter of your sample. So upon the addition of your sample to your working reagent, time for your first minute, make sure that it is placed at 37 degrees Celsius, and then read the absorbance per minute until the third minute. So if this is done manually, we need to compute for your mean absorbance change per minute, and I've already discussed this one. So once you already computed for this, then you need to multiply it by 16,030. So below is your reference range. So that depends on the age. Your LDH is age dependent because, of course, as we all know, when you are still young, your red blood cells are still also developing, or basically all parts of our body are still developing, and thus there is difference in the reference range for your lactate dehydrogenase. So for us adults, that is 225 to 450 U per liter. So for the linearity of this test, so medyo mataas compared to your other enzymes, because diba sa other enzymes, they only have around up to 1000 U per liter na linearity, but here for our LDH, the linearity is up to 2400 U per liter. And in terms of sensitivity, iba din po, mas mababa yung kay LDH, and that is around 0.2 U per liter. So now we go to your phosphatases. So we've discussed this one during our lecture. So just a recap that your alkaline phosphatase and your acid phosphatase are under your third classification of enzyme, and these are your hydrolases. So the EC number for your alkaline phosphatase is EC3131, while for your acid phosphatase is 3132. If you try to look at this slide, the only difference between your alkaline and your acid phosphatase is in terms of its optimum pH. So being an alkaline, meaning to say dapat andito lang po yung range ng pH around 9 to 10. For your acid, that would be at 5. Another thing that you need to take note is in terms of its systematic name. Never ever forget to write this one down. Because if you try to read the systematic name, they are actually just the same. Diba? Orthophosphoric monoester phosphohydrolase. But in order to differentiate whether we are pertaining to your alkaline phosphatase or to your acid phosphatase, you really need to place this one. Naka-open and close parenthesis and then writing down alkaline optimum or acid optimum. The roles of your alkaline phosphatase and acid phosphatase is in terms of catalyzing the hydrolysis of various phosphomonoesters depending on the pH that is needed. So, kung alkaline phosphatase, alkaline, kung acid phosphatase, acid pH. And the cofactor for both are the same. So what they need are your magnesium and your zinc. So looking at their chemical reaction, again, it's just the same. So here, being a phosphatase, meaning to say we are removing a phosphate group. So as you can see here, there's presence of water because again, they are hydrolases and they undergo hydrolysis for your chemical reaction in order to break down certain compounds, and always our end product would be presence of alcohol and then removal of your phosphate ion. So what your phosphatases do is to liberate inorganic phosphate from an organic phosphate ester with the concomitant production of your alcohols. So how about the assays for your phosphatases? So naka side by side po si alkaline phosphatase and acid phosphatase for easy differentiation. So let us begin with alkaline. Now for your continuous monitoring technique, what we do is to calculate your ALP activity based on the presence of your P-nitrophenol, which is actually your product. So your substrate would be your P-nitrophenyl phosphate, which is originally a colorless compound, and it will become your P-nitrophenol upon the addition of your enzyme ALP, now garnering or now having a color of yellow. So, bali kids, the intensity of that color would be directly proportional to the amount of enzyme activity. So, the more intense ang yellow, then that would also mean that there are increased number of alkaline phosphatase in that certain patient's blood sample. So you would get to observe an increase in absorbance, and it is measured at 405 nanometers. Now there are two names of the test indicated here. This is, it actually depends on what type of method we are looking into. For your Bessel-Lowry-Brock, this is an endpoint na method. For your Bowers and McComb, which is a otherwise known as your SAJ modification method, this is a kinetic method. But either way, whether we are using your Bessel-Lowry-Brock or your Bowers and McComb, both have the same inhibitors, and that is your phosphate, your phosphorus, your borate, your oxalate, and your cyanide ion. So since your alkaline phosphatase may be inhibited with the presence of these different substances, what we usually do is we will add a certain substance, and that is what we call your 2-amino 2-methyl 1-propanol. So again, what we will add in our sample, in our testing, is your 2-amino 2-methyl-1-propanol in order to prevent your ALP from being inhibited by the presence of these substances. Now, how about your acid phosphatase? Now, there are two substrates that can be used depending on also on the type of reaction that we wanted to proceed. If we wanted to measure your ACP based on its endpoint reaction, then we will make use of your thymolphthalein monophosphate. If we wanted to measure it through continuous monitoring method, then what we will use is your alpha-naphthyl phosphate. Now, these tests also have their name of the method. For your thymolphthalein, we call this your Roy and Hillman method. While for your alpha-naphthyl phosphate, we call this one, let me just write it here kasi medyo mahaba. That is what we call your Babson, Reed, and Phillips method. So again, these are the names of the method, and I guess these are also the people involved in making this procedure or assay. Now, in testing your acid phosphatase, we can also do other immunochemical techniques like your radioimmunoassay, your counter-immunoelectrophoresis, your immunoprecipitation, and your immunoenzymatic assay, or what we call your tandem E. Now, with regards to your tandem E, what we use here, kids, is a certain antibody that is able to inhibit your prostate as ACP. So just refer to your lecture. We've discussed this one, that there are different isoenzymes also of your acid phosphatase, and one of which is your prostatic ACP. So here in your tandem E, what we will do is we will incubate our blood sample with an antibody that will inhibit your prostatic ACP, and it is followed by washing and certain incubation along with your substrate, which is your p-nitrophenyl phosphate. Now, once your p-nitrophenol is formed, and that will already be directly proportional to your prostatic ACP. So, ganun yung principle and method of your tandem E. So, this is the chemical reaction of your alkaline phosphatase and your acid phosphatase. So, basically, same naman lang din po sila ng ginagamit na substrate. Usually, it is your p-nitrophenyl phosphate, and then the product would be your p-nitrophenol. So again, what differs is in terms of the pH that is needed to be maintained. Now, this test, kids, this reaction here in your ACP is what we call your Shinohara method. As you can see, there are 3 different methods being done, or that can be done rather, with your ACP. So another ACP assay would be in terms of the use of your tartrate. Tartrate is a chemical inhibitor of your prostatic ACP. So what we will do here is, first, we need to measure your total ACP first, after which we will add your tartrate, and then measure na naman po yung ACP activity after the tartrate inhibition. And in order to get the concentration or the measurement of your prostatic ACP, so what we will do is to subtract the value of ACP after tartrate inhibition with your total ACP. But for this one, kids, it is not entirely specific for your prostatic ACP. There are actually far more better tests that we are now routinely doing in the laboratory, and that is your PSA, your prostate-specific antigen, which is a more accurate and a more specific test to measure any problems in your, in order to be able to check if there are any problems in your prostate. So for the sources of error, starting with your ALP. Now, ALP Bawal po hemolyze the sample as it may cause slight elevations in our ALP activity. Your ALP is 6 times more concentrated in your red blood cell than in your serum. ALP should also be processed immediately because activity will increase for around 3 to 10% upon standing at room temperature or 4 degrees Celsius for several hours. So again, general rule yun na dapat i-process agad-agad yung mga samples and that your sample should really be should never be hemolyzed. So another thing is that diet may induce elevations, especially to those individuals who are under these blood groups and who are secretors, 25% higher following a high-fat meal. Because remember, dun po sa ating isoenzymes of your alkaline phosphatase, we also have your intestinal na ALP. And that is greatly affected by the food that we eat, especially if it is a high-fat meal. How about your ACP? Now, serum should be separated from your red cells as soon as possible, meaning to say we need to centrifuge immediately your sample upon receiving to prevent any leakage of your RBC and your platelet, since again, ACP can be found in these tissue sources. So kaya, as emphasized here, there should be no hemolysis. Activity will also decrease within 1 to 2 hours if your sample is left at room temperature. Decreased activity is also a result of the loss of carbon dioxide from your serum, which means that it will increase your pH, making it alkaline. Remember, we need to maintain our environment for acid phosphatase activity na acidic. So, kailangan talaga i-process agad-agad and we actually need to make sure that there is no presence of air in our blood sample. So as much as possible, we need to collect blood in a closed system. So the best thing to do here is not syringe method but your evacuated tube system. So if not assayed immediately, serum should be frozen or acidified to a pH lower than 6.5 because with acidification, kids, your ACP will be stable for 2 days even if it is left at room temperature. Now, in cases when we use radioimmunoassay procedures to measure your prostatic ACP, it requires non-acidified serum samples and it can be stable for 2 days when we place it inside the refrigerator. So for your package insert, this is our principle. So as you can see, with the use of your para-nitrophenyl phosphate, and our product here would be your P-nitrophenol, and then the pH that is maintained here is your 10.2. Magnesium here, kids, is again our cofactor. And then you have here the contents of your R1 and your R2. So same pa rin po yung ating reagent preparation. Na 1:5 pa rin po yung dilution, 1 volume of your reagent 2 and 4 volume of your reagent 1. And same in terms of your reagent storage and stability, it is stable until the expiry date as long as it is stored at 2 to 8 degrees Celsius and protected from light. And as to the reagent deterioration, dalawa pa rin po yung ine-emphasize dito sa package insert natin. Turbidity or precipitation in any kit component will indicate already a deterioration in our reagent and therefore must be discarded. So for the sample, we could either use a serum or plasma sample as long as it is free of hemolysis. And then here, naka-indicate po yung degree ng presence of your hemoglobin, bilirubin, or ascorbic acid na as long as it will not reach this amount, then it will not interfere with our test. So for your test parameters, so ayan po, wavelength is 405 nm, temperature is 37 degrees Celsius, linearity, it can be measured up to 2000 U per liter. 3 times pa rin po ang reading for every minute, every 60 seconds. Your sample volume is 20 microliter and your reagent volume is 1000 microliter. So here our factor, if we are manually doing our test, that would be 2,757. And for the reference range, kids, so men, women, and children have different reference range. Of course, you need to memorize this one. For women, that is around 35 to 104. Sa lalaki, that is 40 to 129. And for children up to 15 years old, that is 54 to 369. And then again, linearity up to 2000 U per L. But in terms of the sensitivity, it can only detect up to as low as 6 U per liter. Okay? So now for your acid phosphatase, this is actually a different brand of reagent, kids. And before, way back, we are actually using this one, your Humalizer. Wala pong ACP sa lab. And since wala din po akong makita na PDF file for the Agape package insert, then let us make use of your Humalizer. So here, in the principle of your ACP, so what we use here is your naphthyl phosphate. And then our end product here is a presence of an azo dye. So if you're wondering what is the meaning of this, so just refer here sa baba. So your FRTR salt is your 4-chloro-2-methylphenyl diazonium salt. And then we have a stabilizer, which is your acetic acid. That stabilizer is to maintain the environment of our processing na acidic. So parang yung acetic acid natin yung nagpapa-acidic ng pH ng ating whole assay. So for your reagent preparation, so here we still need to prepare a working reagent also, but dito po, bale, the way your Humalizer points out how you prepare your reagent is bottle na. Na agad, you'll just have to dispense your buff reagent, placing it to 1 bottle of your sub. So yan po, you might wonder, ayan po, buff and then your sub. So yan yung name ng reagent bottles natin, unlike your Agape na what is indicated is R1 and R2. So for the reagent stability, your reagents are stable up to the expiry date as long as it is stored at 2 to 8 degrees Celsius. Pero ito lang din, kids, once you already mix your reagents and made your working reagent will be stable for 5 days or for 24 hours if nasa room temperature lang. So, medyo ano lang di ay pudning sa inyuhang Humalizer na in a sense na dapat makurot nyo dayon. We really need to be able to consume immediately your reagent once we mixed the whole bottle of it. So for the specimen, kids, naka-indicate na serum lang po talaga yung gagamitin na specimen. And as to your assay, so 405 ang wavelength. And then, pwede po tayo mamili ng temperature if we wanted it at 25 degrees Celsius, at 30 degrees Celsius ba, or at 37 degrees Celsius. So, for the measurement, air blank, pwede na. And then, we will go to the procedure. So, as you can see here in the procedure, meron pong nakasulat na macro and semi-micro. So, depende po siya, literal, na when we are saying macro or semi-micro, sa volume, we are looking at the volume of our preparation here sa sample and sa working reagent. So, it would always be best to make use of your semi-micro so that hindi po agad-agad mauubos yung sample and working reagent natin. So, here in your semi-micro, 100 microliter of sample will be added to 1,000 microliter of your working reagent. And in terms of reading your absorbance, we would only be reading our absorbance twice. Yung first reading po is after 5 minutes. Yung second reading po, depende sa temperature. If we made use of your 30 degrees or 37 degrees Celsius, then it should be exactly after 3 minutes. Or if nag-room temp lang po tayo, then it should be after 5 minutes. And then by the time we already have these two absorbance. So, ganito lang po, pagkuha ng mean absorbance natin, all you have to do is just to really subtract your absorbance 2 to your absorbance 1. So, for the calculation, after getting your mean absorbance change, multiply it by 149 if room temperature po, or 248 if 30 or 37 degrees Celsius. For the linearity also depends on the temperature. If your linearity is greater than 0.3 for 30 degrees or 37 degrees Celsius, for your room temperature, that is greater than 0.5. Or if you wanted to really base it on onto our result na talaga, kids, so it is only linear up to greater than 63 U per liter. So what we will do here is again dilution. Dilute 0.1 ml of your sample with 0.2 ml of a saline, and then repeat the assay and then multiply it by 3 because this is a 1:3 na dilution. So, depende na din po yan sa atin if you wanted to raise the volume, basta as long as kung ano po yung dilution na ginamit natin, then yan din po yung ating dilution factor. So, here in our reference value, iba po sa lalaki, iba din po sa babae, and magkaiba din po in terms of the assay temperature that we use. Basta kay up to man siya, kids, so it will begin with 0 up to ito po na mga levels. So that ends our discussion here regarding your lactate dehydrogenase, your ACP, and your ALP. So thank you so much for listening, kids.